Welcome guys to my YouTube channel Rare Science Simple. Let's make the science simple. I am currently giving lectures on CRISPR and today we are going to cover the topic of designing of single guide RNA. Uh, Suppose how you can design the single guide RNA. Suppose this is the target sequence which you want to modify, and for this, you need to identify the protospacer adjacent motif, which is usually the NGG sequence. In this case, T is the N nucleotide and the GG. So this is basically the NGG sequence. So what the softwares will do, they will uh, create the protospacer. They will design the protospacer for you um, by just observing the uh, PAM sequence in your coding strand. And suppose this is your coding strand and there this is the PAM sequence which is NGG. So they will just count the 20 base pair upstream. So your protospacer is going to bind to the complementary or you can say the template strand here. So let's say this is going to be your template strand and this is your non-template strand. You can say the coding strand, you can say the non-complementary strand, or you can say the non-target strand. Okay, these are the same thing. Similarly, the template strand, you can say the non-coding strand, or you can say the complementary strand, or you can say the target strand. Okay, these are the same thing. So your guide RNA will bind to this template strand, not to this strand. So your guide RNA should have the complementary sequence to this molecule. So to these 20 base pairs. So you can see your sequence is going to be, suppose this is your 3' to 5' sequence. So your guide RNA is going to bind to this particular sequence. Okay, so it will be like GAAC, which is like exactly complementary to this. So it is, the sequence is going to be like here, you can see here. Let me just highlight with some other color here. So it is going to be exactly like this sequence, which is this sequence. Okay, because this is the sequence, if you see here, this is the sequence which is going to be complementary to this template strand. So you will be having GAAC, CGG, CGC, CTA, GAG, AAG, and G. So this is going to be your protospacer or the single guide RNA sequence. This is going to be your sgRNA sequence. It is also known as the protospacer. But remember, in your sgRNA sequence, the PAM sequence should not be there. Your sgRNA should not contain. So the two things which you need to remember. Number one, is coding strand is known as non-template strand, is also known as non-complementary strand, as well as it is also known as the non-template strand. Okay, so these are the same thing. It is also known as the non-target strand. You need not to be confused by all of these synonyms of coding strand. Similarly, the non-coding strand is also known as the template strand, is also known as the complementary strand, just opposite to the coding strand, also known as the uh, uh, target strand. These are the same things. So you need not to be confused by any of these synonyms of coding and the non-coding strand. Third thing which you need to remember, your guide RNA will only bind to your non-coding strand. Okay, this is the non-coding strand. Okay, so your single guide RNA should have the complementary sequence to the non-coding strand, should have the complementary sequence to the non-coding strand. So these are the points which you need to remember. So here you can see this is the non-coding strand, and your protospacer or your sgRNA should have the complementary sequence to this non-coding strand, and therefore we are having this particular sequence because this G will bind to this sequence and like this. So this is your sgRNA sequence. Now suppose in your DNA strand you are not getting the NGG, however you are getting the CCN molecule, so CCN sequence. So in that case, instead of counting upstream, you need to take the downstream bases here. So again, this is going to be your, this is going to be your coding strand, and this is going to be your non-coding strand. And your sgRNA should have the sequence which is complementary to this particular sequence. So here you can see the sequence is going to be like, if you are taking the complementary sequence to this, you will be having C, A, C, G, G, A, T, C, T, C, T, T, C, then you have C, A, C, then you have C, G, C, and G. So if you, because this is the 3' to 5', so when you will order the sequence, you will be ordering G, C, G, C, C, A, C like this. So that is how you need to design the sgRNA sequences. So this is again the sgRNA sequence, and this is the sgRNA sequence which has the complementary sequence to this non-coding strand. Okay, so that's how the softwares also design the sgRNA. For example, this is the CCN molecule. So again, this is going to be the non-coding strand. So any sequence which is complementary to this. So for example, in this case, the complementary sequence is ATT, CA, CT, GG like this. So this is going to be your the protospacer sequence. That's how the softwares also design the sgRNA. But you know, the sgRNA, when you have designed your sgRNA sequence, you need to clone them in the vector, plasmid vector, let's say. So you need to have the complementary sequence also of your sgRNA sequences. So for example, here, if suppose this is the sequence you have designed, you need to have the complementary sequence to this bases also, so that you can clone this sgRNA in the plasmid vector. Okay, so what happened for that? Suppose the plasmid vector which you are using is containing the U6 promoter. So when you will digest this plasmid with the BSM-B1 enzyme, restriction enzyme, this the stuffer fragment is going to be removed. So what is going to be the stuffer fragment here? This is going to be your stuffer fragment. This from here, like this. So this is going to be your stuffer fragment. Okay, so you will be having the overhangs. For example, here you will be having the GTGG, and here you will be having the G triple T, and that is the region when you have designed your sgRNA and the complementary sequence of your sgRNA, you need to add the CACC, which is here you can see the complementary sequence is going to be the CACC here, and that is the reason we are adding CACC. And similarly, the complementary sequence here is going to be, if you take the 5 prime region of this sequence, you will be having the, you need to add in the reverse primer AAC. So what you will be doing here, you will be adding in the forward primer CACC and in the reverse primer AAC, because you know whenever you are ordering the primers, it is always from 5 prime to 3 prime. So this is how this is going to work. Okay, and if you are using the vector that contain the U6 promoter, it's better to add the G nucleotide at the 5 prime position. Okay, so that's how the software is also designed the sgRNA. Now, you know, in case of the homology dependent repair, or you can say the knock-in experiment, where you have modified your gene, instead of just degrading the DNA, knock out the gene, you have just knock in the gene, you have corrected the mutation. So in that case, you can correct the mutation either in just one strand or in both strands. If suppose for the detection whether it has been done in just one strand or in two strand, the T7 endonuclease assay is basically going to be used. What does this enzyme do? So whenever you have done the modification, you isolate the product, amplify that product, denature and reannealed. So what will happen? Then you use the T7 endonuclease enzyme. The characteristic feature of this enzyme is this, that it can cleave only the heteroduplex DNA. Means one strand is modified and the other is not. For example, in this case, it will cleave this sequence, this sequence, this sequence, and this sequence. So if I will write here, I can write here, it will cleave this sequence, this sequence, this sequence, and this sequence. But it will not cleave the sequence if it is not modified or modified on the both strands. In this case, it is not modified in the both strands, this one and this one, and in this case it has been modified in the both strands. So in these three it has not been modified, and in this clone that has modified both the strands. So how you will suppose if only one strand is modified, you will get the full length product and the cleaved products, and you can identify that the mutation has been done in one of the DNA strand molecule. But what if the mutation has been done in the both strands? Then in that case, because the T7 endonuclease only cleave the heteroduplex molecule, it means the molecule that contain the modified DNA at one strand and unmodified DNA at the another strand. So this, because this is not this has been modified, this particular clone has been modified in both the strands, T7 endonuclease will not cleave. And similarly, these strand which has not been modified in both the strands, and T7 endonuclease will not cleave this either. So how will you recognize these clones? So for that, what you have to do, you need to add the sgRNA molecule. Suppose this is your clone that has the modified DNA at both the strands, and now you add the single guide RNA that has not been modified. So what it will do, it will react with this particular strand, and then it will form the heteroduplex molecule. And once it will form the heteroduplex molecule, the T7 endonuclease will now cleave this, because it is heteroduplex, and therefore you can recognize that your clone has both the strands that has been actually modified. Okay, so what you have to do, you have to add the RNA sequence which has no modification. These are the basically softwares which are used for designing the sgRNA. And this one, the chop is very good software that you can use for designing your sgRNA. And similarly this sgRNA designer, so number 5 and number 8 are the good softwares which you can use for designing your sgRNA. So that's all about this lecture, and if you like the content of my lecture, kindly subscribe my channel. Thank you so much.